Good morning students, welcome to Leela's tutorial. Today we will discuss about another technique, that is RAPD, randomly amplified polymorphic DNA technique. So this is one of the molecular marker technique. We have three different types already we have discussed in last class, that is first generation molecular markers, second generation and new generation. So we have discussed about RFLP and now this is the second type, this is RAPD. These two come under your first generation molecular markers. So here normally molecular markers we use to identify and differentiate one of the organism from a number of organisms or the animals. So let us see the technique. First, mainly it is PCR based technique. That means here you are making use of these markers with the help of PCR. In PCR, you can amplify the DNA fragments, right? You can make more number of copies, right? So it is mainly based on PCR technique. Then second point is it is using arbitrary primers. That means these are small synthetic primers, right? They are synthetic primers which are prepared, right? Then the another thing is they require the non-specific sites. In RAPD it is to the specific site we were producing complementary primers, whereas here the arbitrary or the synthetic primers are randomly prepared. You are not preparing them something complementary to the DNA, right? So you are preparing the primers and you don't know to which site they go and bind, right? So that means you don't know even the what is the genome sequence or the analysis of that particular animal, right? So here no specific sites are required, right? Randomly you will be taking the DNA from the animal and you will be adding these arbitrary primers so that they go and bind to the DNA randomly at different places, right? To the complementary locations. So another thing is amplification. Already we had told that it is PCR based technique. That means after binding of these primers to certain sites, what happened? They undergo PCR and they produce more number of copies. That is referred as amplification of that particular DNA fragments. The next one is, after that you are going to run the agarose gel electrophoresis. Okay, you are allowing the fragments, DNA amplified fragments to migrate in the agarose gel so that they form different band patterns. Okay, so let us see the complete technique here in the diagrammatic representation. So first, here I am taking sample A from one animal and sample B from another animal. So here first step will be, I will be taking the cells, right? Then the cell lysis, then after cell lysis it is isolation of the genomic DNA. So once I isolate the genomic DNA of sample A and sample B cells, then next step is adding of primers and DNA polymerase. So here arbitrary primers must be prepared in prior, right? So these are small synthetic primers. See, now you have this double stranded DNA, right? 5' to 3', 3' to 5'. Okay, complementary bases. So here complementary strands. So these are the sample A DNA and this is the sample B DNA. So to these DNA, I am going to add separately the primer and the DNA polymerase. Even here the primer and the DNA polymerase. So once I add these primers, they go and bind to their complementary sites, right? You don't know how many sites are present, right, in this genomic DNA where these primers will go and bind, right? So randomly you are adding, so these primers which are added, they go and bind at that particular complementary sites, right? They go and bind for both A and also for the B. So here in the diagram I have shown that you have how many sites here for the primers? It is 3 sites are present to which your primers will go and bind, right? And again in the same way to the sample B genomic DNA. The primer I have shown the two sites to which they go and bind. Okay, so this is double stranded DNA. So these are the sites where they can go and bind. So the 4 primers will go and bind at the complementary sites. And what is the use of DNA polymerase? They allow the fragments or the sequence to extend. Okay, they move from 3' to 5' end, so that they will elongate to form a DNA fragments. Right? So see here, I have shown from the 3' to 5', and these are the sites where the primers go and bind. You can see here the primer has gone and bound to this. Right? Then the red dotted line is the extension or amplification process. Right? The complementary strand is being synthesized. In which direction? I have shown the direction 3' to 5'. In the same way, the second strand also you can see the 3 sides where the primers are bound and they are resulting in extension of the fragment, right, in 3' to 5' direction. In the same way, here you have 2 fragments, right, 2 sides. So 4 fragments are formed. So here it is a PCR based where the amplified, see this particular fragment or this particular region is being amplified because of this. Right? So like that, so many copies are formed. Isn't it? You are going to produce so many copies by using your polymerase chain reaction technique. Right? Now you got amplification by RAPD. Okay? Rapidly amplified polymorphic DNA. Right? Or sorry, randomly amplified polymorphic DNA. So once you have got these amplified fragments, then what you do? You are going to collect these, right? And you have to migrate these amplified fragments of A and B in agarose gel electrophoresis. Right? So here in the agarose gel, you have well A and well B. So A sample, amplified fragment sample will be loaded in this and B will be loaded in this. Right? Then allow the fragments to migrate in agarose gel. As a result, you can observe the band pattern. Right? That means here the 4 bands are formed, whereas here 3. That means what? Here in the complete genomic DNA, for suppose I can say that the primers which are produced, right, the synthetic primers which are produced, they have gone and bind to certain sites and resulted in formation of these 4 different sized fragments. Right? So now you can see the band pattern is different for animal A and also for the animal B. So like this you can compare them and also compare the size of the amplified fragments. See, the smaller sized DNA fragments migrated long distance, whereas short distance is for the longer size or large size DNA fragments. So here the RAPD mainly helps in detection of size of amplified fragments and also for the comparative studies between different animals. Is that clear? So then you have other advantages. So what would be the first advantage? You doesn't require any specific locate. Right? You don't want any specific site to be identified. Right? You can produce a synthetic primer as such. Right? Then second one is amplify low DNA fragments. Very less sized DNA fragments can be produced by using your RAPD technique. The next one is no sequence information. That means we are preparing the primers as such. We are not preparing them specific to the site. If you are going to produce the primer specific to certain site, then you must know the genome or gene mapping of that particular genomic DNA. Isn't it? You have to know what are the genes present, what is the site to which you are going to produce the primer. Right? But here in this technique, you need not know any information about the genomic DNA because you can use any synthetic primer randomly. Okay? Then next one, they show a codominant alleles. So these are the advantages of this RAPD technique. If you have any doubts, put it in comments and subscribe for further videos.